Hello friends, welcome back to Smriti Notes for Biotechnology. So in today's video we will study about what are antigens, antibodies and about their types and interactions. This is the basic structure of the antigen and the antibody. Now first we'll study what are antigens, what are antibodies, and then we'll study the interaction process between them. So an antigen is a substance that reacts with the products of a specific immune system. That is, it is a substance that causes an immune system to produce an antibody against it. That is B cells. Antigen is a molecule that binds to antigen specific receptors. Antigens are usually peptides, polysaccharides, or lipids. Epitopes are components of antigens. Each antigen carries many epitopes. Each Y-shaped antibody molecule has at least two binding sites that can attach to specific epitope. This way, the one side and the other side. Now these are various properties of antigens like immunogenicity, antigenicity, allergogenicity, and tolerogenicity. Now they are of various types based on their origins. The very first one is exogenous antigens and the other is endogenous. Exogenous antigen means that the antigens have entered the body from outside, like by inhaling, ingestion, and injection. Now the next type is endogenous. That means the antigens that have been generated with previously normal cells as a result of normal cell metabolism. So this was all about antigens. Now we'll study about antibodies. They are also known as immunoglobulin. It is a large Y-shaped protein produced mainly by plasma cells and is used by immune system to identify and neutralize pathogens. Antibodies are secreted by B cells of adaptive immune system. Antibodies are glycoproteins. They are typically made of basic structural units, and each unit has two large heavy chains and two small light chains. We'll study about heavy chains and light chains in detail. Heavy chain. The heavy chain gene locus shows basically two segments. They are variable and constant segments. I'll show you the diagram also. The variable segment is basically made up of three loci, that is V, D, and J. Now there are five classes they are given here. All polypeptide contain tandem series of segments, and these segments are homologous. That means they are all same. The N-terminal variable region and C-terminal is the constant domain. The type of heavy chain present defines the class of antibody, and these chains are found on IgG, IgA, IgD, IgE, and IgM antibodies. Alpha and gamma contains 450 amino acids, whereas these contain only 550 amino acids. Now, what is the hinge region? You know hinge, what is a hinge? Hinge is a place where two joints come and meet. So basically, flexible amino acids stretch in central part of the heavy chain of IgA and IgG immunoglobulin classes, which link these two chains by disulfide bond. Gamma, delta, and alpha chains have hinge region, no resemblance to any other Ig regions. This is the overview of how the attachments are done. This is just the arrangement. We'll discuss about the arrangements in the next video. Now, as you can see, DJ arrangement, VDJ arrangement, and all. That is how they are linked through the hinge. Now, let's study about light chains. And the light chains are like this. This is the arrangements of light chains. In mammals, there are two types of Ig light chains, which are called as lambda and kappa. They have two successive domains, that is one constant and one variable, and their length is about 211 to 270 amino acids. Now they have various isotopes. I'll be showing you. If you guys want to write them down, you can just copy it. The first one is IgG. They are basically present in the new bonds. I've mentioned some other details as well here. You can pause the video and write it, and if you're not able to understand any of the words, then you can just drop me a message in the comment box. I'll help you out. The other type of isotope is IgM. As you can see, The next one is IgA. I'll just focus it for you guys. The next one is IgE, and then is IgD. These are the types of isotopes that are present in light chains. So in this video we studied about light chain, heavy chain, which are a part of it, and also about antigen and antibodies. In the next video I will be teaching you about the interaction. So guys, if you like the video, then do not forget to like, share and comment. Antigen antibody interactions. The antigen and the antibody combine specifically with each other. As we have seen in the previous video what antigens and antibodies are, now they both are combining, and the interaction between them is called as antigen antibody reaction. They form the basis of humoral immunity or antibody mediated immunity. The reaction form the basis for detection of infection, disease causing agents. So basically when we are having any specific kinds of disease or infection, we can find out through antigen antibody interactions. When antigen antibody reaction occurs in vitro, they are known as serological reactions, and the reactions between them occur in 3 stages. And the first stage is formation, that leads to visible events like precipitate agglutination, and the last one is the destruction of antigen. Now we'll look at the applications. Antigen antibody interaction is used in laboratory techniques for serological tests, that is for blood compatibility, and they are the basics in the ABO blood group determination. Antigen antibodies. Now we look into the features of antigen antibodies. The very first is the specificity. Some of the features of antigen antibody reactions are specificity of antigen, that is the ability of a population of antibody molecule to react with only one antigen. The next is immune complex, that is they both combine to form the antigen antibody complex. Now the binding site of antigen, it basically attaches with the antigen, and the part of the antigen while combines with antibody is called the epitope. The binding force of antigen is due to the closeness between antigen and antibody, non-covalent bonds, and the affinity to antibody. This was features. Now we look at some of the properties. That is, the first one is antibody affinity. What does this mean? It is the sum of the attractive and repulsive force. That means for antigen and antibody to come together and to get repelled, the force operating between the antigenic determinant and the combining site of antibody. The higher the affinity of the antibody for antigen, the more stable will be the interaction. This is the equation which I have given. Now, the next is avidity of antibody. It strengthens the bond after formation of antigen antibody, and they devote the overall capacity of antibodies to combine with multivalent antigens. Now, the last feature is cross reaction. That means ability of an individual antibody combining site to react with more than one antigenic determinant site of antigen. It occurs when 2 or more antigens share similar structures, and these bonds involved are in cross reaction are weak. The bonds are very weak. So this was about the antigen antibody interaction. So if you are able to understand, and if you have any doubt or you're not able to understand any word, you can comment down in the comment section and let me know. And if you're finding these videos useful, then do like, share and subscribe to my channel.